I'm talking about cellular rejuvenation and potential immortality. I'm also talking about how cancer cells cheat, because I think that cancer cells somehow manage to reactivate a potential immortality system that is normally lost in the cells of animals. Now, this is a subject I've been interested in for a very long time, although curiously this is the first talk I've actually ever given on the subject. In the 1970s, I was working on cell biology at Cambridge, and particularly interested in the whole question of cellular senescence and rejuvenation. And in 1974, I published a paper in Nature called The Aging, Growth and Death of Cells, setting out a new approach to understanding aging and rejuvenation at the cellular level. And this has been a subject that's remained of interest to me ever since. And in 2022, I published another paper on the subject, bringing the whole thing completely up to date and putting forward a new version of my hypothesis in this paper called Cellular Senescence, Rejuvenation and Potential Immortality, published in the Proceedings of the Royal Society B. B means biological. What I'm doing in these papers is looking at two paradigms of aging and rejuvenation that have dominated biology since the late 19th century. And they've coexisted because they coexist in different subfields of biology, even though they're incompatible with each other. And I think that this long debate or rather disagreement between these two paradigms can actually be resolved. And I'm going to suggest in this talk how it can be resolved and how its resolution could shed a new light on cancer and possibly even lead to new ways of treating cancer. The debate started in the late 19th century, in the 1870s and 80s, and it was between two eminent biologists. One was called Émile Maupin, who was French, lived in Algiers, and studied many forms of organisms, small organisms. He was the first person to name the small nematode worm Caenorhabditis elegans, which is now one of the main model organisms in biology. The other was August Weismann, a German professor of biology, who had an enormous influence on the development of genetics. Maupin argued that all cells age all the time, that aging is inherent in life, that things go wrong, problems build up, junk accumulates, that aging is inherent in all life. So for Maupin, the question was, how does anything ever get rejuvenated? Because if aging went on in all cells all the time, all life would die out. So there has to be a rejuvenation process. And he thought that happened through sex. He wasn't quite sure how, but he had the idea that sex is the rejuvenative process that enables new generations to be regenerated. His main experimental studies were on a small unicellular organism called paramecium, a ciliate that swims around by beating its little hairs on the surface. And these do indeed age, colonies of paramecium age, and they are rejuvenated by sexual conjugation. So he had good evidence that this was actually happening. His opponent, August Weismann, took the opposite point of view. He thought that cells, single-celled organisms, are potentially immortal, that they can just go on and on and on without showing any signs of aging, and that aging only happens in multicellular organisms. The germ cells, especially the egg cells, continue with this potential immortality that they inherit from unicellular organisms. but other cells age. As he put it, he said the animal germ cells inherit their potential immortality from unicellular organisms which, as he, in his own words, carry the potentiality of unending life. The reproductive cells cannot lose that capacity for unlimited reproduction, but the somatic cells have lost that power. They become restricted to a fixed, though perhaps very large number of cell generations. So he had the idea that germ cells are special, unicellular organisms are special. They have a capacity to rejuvenate themselves all the time. What needs explaining is senescence. So these two paradigms, which I call the universal senescence paradigm and the potential immortality paradigm, are almost opposite. For one of them, the problem is to explain rejuvenation. For the other, the problem is to explain senescence. Now, the potential immortality paradigm still dominates areas of biology concerned with egg cells and also cancer, because many cancer cells have acquired a kind of potential immortality. However, the cellular senescence paradigm, the universal senescence paradigm, is now predominant in many parts of biology, including animal development studies, and also in the study of yeasts and bacteria. It turns out that unicellular organisms do indeed senesce and age, as Mopar has said and as Weissman denied. When I got interested in the subject in the 1970s, the dominant theory was the potential immortality theory. And there were attempts to explain senescence in terms of cascades of errors, like the so-called error catastrophe hypothesis of Louis Augel, and a subsequent theory, Kirkwood's disposable soma theory, explicitly took up Weissman's ideas. He assumed that the germ cells were potentially immortal, but that it was necessary to explain why other cells age. I myself, when I got interested in this, started from the universal senescence paradigm, the idea that all cells age all the time. And I was trying to work out how cells can be rejuvenated. I put forward a hypothesis that made sense to me at the time and has since been widely confirmed. This hypothesis is a very simple one, that as cells get older, they accumulate damaged cell constituents. Cells have very good repair systems. They can get rid of denatured proteins. They have protein dissolving systems called proteasomes that enable them to recycle dysfunctional proteins. They have repair systems for membranes. They have repair systems for errors in DNA. They have wonderfully effective repair systems, but there's always some things these systems can't repair. And what happens is that indigestible residues, bits of undigested protein or mitochondria, accumulate in the digestive vesicles within cells called lysosomes that dissolve material that otherwise needs to be digested. There's undigested residues. These accumulate within cells, as do damages within cell membranes. I call all these damaged cell constituents. So the damaged cell constituents build up in cells as time goes on, and if cells don't grow and divide, then they're eventually overwhelmed. They senesce because of the build-up of these damaged cell constituents. However, they can get rid of them by growing and dividing. If they grow, they dilute them, and if they divide, they pass them on to their daughter cells. And there's two main ways in which cells divide, as I show in this diagram. The symmetrical pattern of cell division means that the cells divide to give two more or less equal cells, and those divide to give, each of those divides to give two more or less equal cells. And if there are damaged cell constituents, these get shared between the two cells, so they both inherit some damaged cell constituents, and more build up as time goes on. Then their daughters inherit more than they themselves inherited if they continue to build up, which they usually do. So everything depends on how fast they divide and at what rate these damaged cell constituents build up, and also whether the damage is autocatalytic, whether damage leads to more damage happening at a faster rate. And as a result of my paper in Nature in 1974, a whole school of mathematical modeling grew up, which showed that if you vary these parameters, these cell population will die out more or less quickly if they divide symmetrically. So everything dependent on these rates of division and accumulation. But the other way in which cells can divide is asymmetrically. And as I show in this diagram, when they divide, they could divide so one of the daughter cells receives most or all of the junk of the damaged cell constituents, wiping the slate clean for the other one. So one cell is rejuvenated at the expense of the other. And this means that the rejuvenated cell, it will still accumulate damaged cell constituents, but starting from a clean slate, more or less, when it divides, it can get rid of them again and can keep doing this and keep being regenerated, giving rise to mortal daughter cells. So one of them, as it were, inherits the seeds of mortality, and the other one is regenerated through, one of them takes on this burden of mortality to regenerate the other. This can happen in two ways. Either the mother cell can pass on these damaged cell constituents to a daughter cell and then keep regenerating the mother cell. The daughter cells are then all mortal. Or it can happen the other way around. The daughter cells can be regenerated while the mother cell retains all the damaged cell constituents, in which case the mother cells will gradually age and eventually die. And we find, in reality, both these patterns. In embryonic animals, when the mother cells are giving rise to nerve cells or to egg cells or to other cells that are important for the growth of the embryo, the mother cells tend to retain the damaged cell constituents, and they're disposable anyway because they're just helping to build up the embryo and giving rise to new nerve cells. So it's the mother cells that retain all the damaged cell constituents. But in stem cells in our own bodies, for example, the stem cells that give rise to new skin, the stem cells that give rise to new blood cells within the bone marrow, the stem cells that give rise to new intestinal lining, these stem cells keep dividing, keep giving rise to specialized cells like skin cells, which die as they differentiate. The intestinal lining cells only last a few hours or a few days. The blood cells only last for a limited period, then they die. They don't need to be immortal, but the stem cells keep being regenerated, having their slate wiped clean every time they divide. And so in our own bodies, the production of new cells largely happens by stem cells, and the stem cells are regenerated, rejuvenated by the process of division. It's now been studied in considerable detail how this process works, and when I hypothesized this in the 1970s, no one could actually see how this happened. You couldn't actually study the details of cell division, but it's now become possible to do that using labeling techniques. And here, for example, is a study of stem-like cells in human mammary epithelial cell cultures. In this case, the mitochondria, which are small organelles within the cell that produce energy, were labeled so that older mitochondria were labeled so they glowed red, and younger mitochondria glowed green. And when this cell divided, when they divided, the older mitochondria largely went into the cell which died, whereas the stem cell itself was regenerated. It got very few of the old mitochondria. It got a higher proportion of the young mitochondria. Now, it's also been shown through using fluorescent labels and other ingenious methods that denatured protein aggregates and all sorts of junk within the cells is asymmetrically partitioned during the division of stem cells, regenerating the other cell. So what started as a hypothesis is now a well-established fact in many areas of animal biology. Cells are indeed rejuvenated by passing on damaged cell constituents, regenerating the stem cells. In plants, there's a similar situation. These stem cells in the meristems, the growing points of the shoot tip and the root tip, divide asymmetrically to give one cell that remains a stem cell, a meristematic cell, and the other cells that can undergo further division, but then they become wood cells or bark cells or leaf cells. And all these cells die sooner or later, and yet the meristematic cells can go on being regenerated indefinitely. In a tree that's 2,500 years old, for example, you can take cuttings from the stems, from the meristems, from the ends of the stem tips, and you can grow a whole new tree from these cuttings. There's no aging in these cells. They're continually rejuvenated. And that, I think, is happening because of asymmetric cell division and passing on the damaged cell constituents. People have not actually shown this directly in plants yet, but I predict that this will be found when people look for it. In my Royal Society paper, I have an appendix suggesting some new experiments, and one of them is applying the same techniques that have been applied to animals to plant cells, and I think the same pattern will emerge. Fungi may do the same thing. Multicellular fungi divide when they produce spores in such a way that the spore from which the new organism will come has the slate wiped clean, and the damaged cell constituents may well be partitioned into cells that remain and sooner or later die. Again, this hasn't been examined directly, but it could be quite easily, and so this hypothesis could be tested in fungi as well. It's already been tested in unicellular fungi like yeasts. Yeast cells come in two kinds. One kind of yeast is called budding yeast, and what happens there is a big yeast cell gives rise to a little bud cell, which then splits off and goes away, and it becomes a free-living cell itself. The mother cell, which buds off a whole series of daughter cells, in the case of yeast, accumulates damaged cell constituents, and after a given number of generations slows down in its division, and eventually they die, whereas the cells they give rise to become mother cells themselves and bud off cells until they accumulate lots of damaged cell constituents and die. At first, people thought that the other kind of yeast, called fission yeast, which divides symmetrically, giving two equal cells, were somehow very different. It now turns out that this symmetry is illusory, that actually one of the cells is inheriting the damaged cell constituents and ages and dies, and the other cell is rejuvenated. So there's a mixed population in fission yeasts, the ones that divide symmetrically, where some of them, even though they look the same, are actually accumulating these damaged cell constituents, aging and dying, while others are being rejuvenated. And the same happens in bacteria. There are some bacteria that divide asymmetrically. There's one called Caulobacter, which is an aquatic bacterium, and the top picture in this diagram shows a Caulobacter. It has a cell which has a stalk anchoring it to the medium on which it sticks. and it buds off a flagellated cell, a cell that can swim away, and the stalked cell retains the damaged cell constituents, regenerating the flagellated cell, which can then become a stalked cell itself, and, like a budding yeast, give rise to new cells while itself ages. It ages itself, regenerating the daughters. But many bacteria, like the famous E. coli, used so widely in biological research, divide symmetrically. And like fish and yeasts, at first people thought that this meant that any damaged cell constituents would be symmetrically distributed. But that's not the case. As this diagram shows, one cell wall at the end of the cell is older than the other, and damaged cell constituents accumulate near the older cell wall. So one of the cells inherits them, the other doesn't. And the one that inherits them divides slower and ages, while the other one is rejuvenated and divides quicker. And every time they divide, there's this asymmetry, meaning that some of them are getting older, bearing the burden of senescence, while others are rejuvenated within the population. So this pattern of the asymmetric distribution of damaged cell constituents is found very widely in unicellular organisms, probably in plants, and certainly in many animals and in yeasts. And all this fits with the hypothesis of rejuvenation through asymmetric cell division. But things get more complicated when we consider cells that divide symmetrically. The symmetrically dividing cells, if they don't divide very fast, and if there's a build-up of damaged cell constituents, would only divide for a certain number of generations before they die out. And it's well known that some cells that divide symmetrically from mammals, such as humans, cells called fibroblasts, for example, that give rise to connective tissue, divide symmetrically and die out after a fixed number of generations, rarely more than 50. And if you take them from older organisms, they undergo fewer divisions before they die out than if you take them from younger organisms. This could be due to the build-up of damaged cell constituents. It's complicated, however, by the fact that there are other factors in the organism controlling cell division as well. The fact that they only divide for a limited number of generations was discovered by a scientist called Hayflick, and so this is sometimes called the Hayflick limit. It's not just due to the build-up of damaged cell constituents. These cells have extensions at the ends of chromosomes called telomeres, and when they divide, the telomeres are shortened. Every time they divide, there's a bit chopped off the end of the telomere, and so when they get down to a certain very short telomeres, they stop dividing. And there are enzymes called telomerase, which extend telomeres. If you extend their telomeres, they can start dividing again. Similarly, DNA and histones associated with DNA can be methylated, a chemical change that is associated with aging. And this, as they get more and more methylated, they divide slower and slower, and this is one sign of senescence. But there are enzymes that can demethylate them, and then they're rejuvenated. So these controls over proliferation through telomeres and through methylation of DNA and histones are one factor in aging. But these cells would still age even if you stop them from aging because of telomeres and methylation. They're still not immortal. There's the aging, the accumulation of damaged cell constituents will still overwhelm them in the end. So how is it then that there are some cells, like cancer cells, which divide symmetrically and yet go on dividing indefinitely? A certain kind of cancer cell isolated from the cervical cancer of an African-American woman called Henrietta Lacks so-called HeLa cells, were first isolated in 1951, and they've been proliferating by symmetrical division in laboratories ever since. And so have many other cancer cell lines, which are called transformed cell lines. Somehow, these cells have acquired the ability to proliferate indefinitely without dying out. So how are they doing it? This seems to be a violation of the basic principle of universal senescence paradigm. It suggests that these cells somehow have a way of rejuvenating themselves. They really do seem to have a kind of potential immortality, just like Weizmann supposed in the late 19th century. Yet, how can this be if senescence is a universal principle? This was a problem that I was wrestling with in the 1970s, and I'm still wrestling with today, but I think it's becoming clearer how there can be a solution to this. The immortality of these cancer cells is not unique to cancer cells. The potential immortality is shared by some of the earliest cells that occur in mammalian embryos, called embryonic stem cells. Embryonic stem cells divide symmetrically, and if you grow them in tissue culture in the laboratory, you can get them to grow indefinitely, symmetrically, without showing signs of aging. What's happened with these cancer cells is they've somehow recovered the properties of embryonic stem cells. In fact, what are often called oncogenes, cancer genes, like a gene called c-MYC, letter C hyphen MYC, c-MYC gene, was for many years considered to be a diagnostic feature of cancer, a kind of mutation that led to cancer. A lot of research on cancer is focused on genetic changes. And by calling these oncogenes cancer genes, it sounds as if cancer is just a genetic problem, and there are special genes that become activated in cancer that make cells cancerous. But it turns out that actually these oncogenes are mainly genes that are normally expressed in embryonic stem cells. And so the key to cancer seems to be the key to understanding what's happening in these embryonic stem cells. And embryonic stem cells exist in normal embryos, like human embryos. They only exist for a few days. Soon after fertilization, as the fertilized egg is trundling down the fallopian tubes towards the uterus, these embryonic stem cells are dividing rapidly to produce the cells which give rise to the embryo. They divide symmetrically and rapidly, and within a few days they become normal stem cells and start dividing asymmetrically and give rise to the cells of the embryo. But artificially, they can be kept going in tissue culture in the laboratory. Interestingly, they have a number of features in common with cancer cells. Embryonic stem cells develop in very low oxygen conditions. They're not in the middle of a well-oxygenated system. They're in a cell going down the fallopian tube. They have only about 2.5% oxygen compared with about 20% in a fully oxygenated tissue, which is with a level in the atmosphere about 20% oxygen. They get their energy mainly by fermentation. And interestingly, it's been known for decades that cancer cells get their energy mainly by fermentation, like embryonic stem cells. You need a lot of oxygen to get it by respiration, which is the normal way we produce energy. And many people have been puzzled by the fact that if you deprive cancer cells of oxygen, instead of doing worse, they do better. If you try and deprive them of a blood supply in tumours, for example, which was one approach to cancer therapy, the cells actually prefer low oxygen conditions because they get their energy by fermentation that doesn't need oxygen, and they're better adapted to this. In other words, it seems to me and to a number of other researchers that one of the key features of the cancerous transformation is to hijack features of embryonic stem cells, which are normally shut down when a normal embryo develops. So in that sense, the cancer cells are cheating. They've found a way of becoming potentially immortal by taking on this brief period of potential immortality that occurs in normal embryonic growth. But this pushes back the problem one stage further. How is it that embryonic stem cells are able to divide symmetrically, indefinitely, if put in the right conditions in the laboratory, and so are HeLa cells and other cancerous transformed cells? How is it that they're able to do this and not be overcome by senescence and not be overcome by damaged cell constituents? It's very improbable that they don't make damaged cell constituents. These happen all the time as a result of wear and tear and cellular damage. And it's very unlikely they have perfect repair systems, because none of the cell's repair systems are perfect. So how do they get rid of them if they're not doing it by asymmetric cell division? Well, in the 1970s, it seemed to me that the only possible way they could do this is by secreting them, by excreting them, forming little vesicles, and pushing out the damaged cell constituents, excreting them into their environment, instead of letting them build up inside the cells. But at that time, very little was known about vesicle formation by cells. There's now been a lot of research on the formation of secretory vesicles by cells. And there are several kinds, as this diagram shows. One kind of secretory vesicle, called exosomes, are secreted into vacuoles, or spaces within the cell with a membrane around them. Then that vacuole fuses with the cell membrane and excretes the exosomes. Some are budded off directly from the cell membrane, called microvesicles. Sometimes viruses are budded off from the cell membrane. They're called enveloped virus vesicles, like HIV, for example. And sometimes cells secrete, for example, fat droplets, like in the mammary gland, in vesicles by budding them off from the membrane. So there are quite a few ways in which cells can actually get rid of things within them, putting them out through little vesicles into their environment. And studies of these vesicles have shown that they contain all sorts of things, sometimes DNA, RNA, misfolded proteins, denatured proteins, and waste products, damaged cell constituents. Now, the hypothesis I'm putting forward now, which hasn't been tested but which seems to me very probable, is that when cells undergo cancerous transformation, or when they become embryonic stem cells, they secrete lots of vesicles and excrete damaged cell constituents in these vesicles into the environment around them. Now, it's actually known that embryonic stem cells secrete lots of vesicles, and some people even try to collect the vesicles and use them for embryonic stem cell therapy, using the vesicles instead of the embryonic stem cells in a form of stem cell therapy. It's a whole field of research looking at these vesicles now. Cancer cells also secrete heightened numbers of vesicles. When they're transformed to being cancer cells, they produce many more vesicles than they did before. And there are now blood tests for cancer that look for these vesicles in the bloodstream from blood samples. These are advanced cancer diagnostic methods that are considered very promising by many researchers at present. So it's plausible that these embryonic stem cells and cancer cells could be excreting damaged cell constituents into the environment around them and rejuvenating themselves by doing so. What I'm suggesting is that in embryonic stem cells, this form of excreting waste products is a temporary phase. The embryonic stem cells are growing within a fluid-filled cavity called the blastocele, which is like a rubbish dump where they can dump these damaged cell constituents excreted in vesicles. But when the cells become part of a normal organism, I think this secretory system is switched off. Because if all the cells in our body were dumping all their waste into the environment around them, the intercellular environment, the whole body would become like a kind of open sewer, and the cleanup systems, the phagocytes that mop up damaged cell constituents, would be completely overwhelmed. So when cells become normal cells within a growing embryo, I'm suggesting they now switch to keeping their waste products within them, and unless they dilute them by growth and cell division, especially by asymmetric cell division, the cells inevitably age, which is why all cells that stop growing age, all organisms that stop growing age. That's why animals age, they stop growing. Plants don't age in the same way because they keep growing. A tree can grow for hundreds or thousands of years because it keeps growing and keeps regenerating those cells. Now, I think that when cancer cells are activated as cancer cells, they recover this ability to rejuvenate themselves, inherited from embryonic stem cells, by, in a very antisocial way, ejecting their wastes into the environment around them. And of course, cancer cells are essentially selfish. They've found a way of escaping from the normal controls of cell proliferation. That's the first step in cancerous growth. Some of them develop from stem cells by becoming more like even like an exaggerated stem cell that just keeps dividing and dividing and dividing. Most cancers originate, in fact, from stem cells. And the second stage in their transformation is this acquisition of potential immortality. And that, I think, is by reactivating these embryonic stem cell systems and secreting their damaged cell constituents in vesicles. Now, if it were possible to suppress the vesicle secretion by cancer cells, then it might be possible to find a new approach to cancer therapy, because the cancer cells would then age like other cells, and instead of being a problem of proliferating indefinitely, they would undergo an aging process and die out by themselves. Now this, as far as I know, has not been explored as an approach to cancer, because so much of the focus has been on genetic changes and changes in proteins and changes within the cells. If there is this system in cancer cells and in embryonic stem cells, it seems to me quite possible that many unicellular organisms, like amoeba, could actually excrete damaged cell constituents as well. They live in fluid environments, in fresh water or in fluids, other fluids environments, where these damaged cell constituents can be washed away and diluted. That's not the case, as I said, when cells are crammed together within plant or animal bodies, but when they're free-floating, they could excrete them, and amoeba is known to have an excretory system. It's possible that it excretes its own damaged cell constituents as well as food residues through that excretory system. So it's possible that some cells are indeed potentially immortal, just as Weismann supposed in the 19th century. Some unicellular organisms, possibly egg cells, which do secrete vesicles as well, and cancerous cells have this ability to be potentially immortal, like Weismann said. But like Maupas said, it's not because they don't age,